So my name is Charles Keller. I'm the scientific director of the Children's Cancer Therapy Development Institute. This is kind of a novel, a 501c3 nonprofit biotech. And our mission, of all things, right? So why should biotechs be nonprofit? But our mission is to move basic science discoveries to the start of clinical trials for children with cancer. Are you guys getting echo on this? No? Okay, it's just me then. All right. So that voice in the head on a 3 millisecond delay. So I'm going to talk today about our Pediatric Cancer Nanocourse for families, essentially a one-week boot camp for families and patients affected by childhood cancer. I'll start out by talking about our motivation. So the fabulous thing is that childhood cancers have a terrific cure rate. It's now at 83%. Adult cancer has a roughly 66% cure rate. And so in pediatric cancer, we've been very pleased to be able to help the 4 out of 5 kids whom we can cure. But there are still 1 out of 5 kids who have the same diagnosis that they've always had that we know won't be curable. And so children like Shane here are our motivation. So as an academic pediatric hematologist oncologist turned basic scientist, I often got questions about my publication record, possibly about what kinds of grants I was going to have. But the hardest question I ever had was from my daughter, who asked, is there anything you've ever done in your life that has ever saved anyone's life ever? And so you can get a fourth percentile R01, you can publish in Cancer Cell Genes and Development, Nature Medicine. But if you can't answer the question from your 4.5-year-old daughter in an honest way, it's time to sort of look for other options. And so if we wanted to help those 1 in 5 kids, for myself, historically, I look back at other models. And so there's 3 pictures up here of people who've changed history. So my favorite scientist, the best scientist that I can possibly think of, had the laboratory on the left, or on your right. This is actually in Paris. It's a warehouse where they sifted through embers to find radium. Does anyone know whose lab that is? Marie Curie. Heard that exactly. Invented the light bulb, among a number of other things. Right. Menlo Park, Thomas Edison. And finally, a self-educated South African computer hacker, code hacker, who thought that banking should be online, who thought that we should power our cars with electric motors and then go to Mars. Whose laboratory is that? Elon Musk. So that's his Dragon capsule, SpaceX Dragon capsule. So the question was, how can we do this? And if we were going to do something revolutionary to help those 1 in 5 kids, what would we do? And what we knew for sure was that we were having plenty of basic science discoveries. We were absolutely positively understanding the targets of disease and childhood cancer, but we weren't necessarily getting those discoveries into clinical trials. And so there was a preclinical gap. I think this was described very well by the Noah's Light Foundation in this video that went viral. It's had 304 views on YouTube. I've watched it 270 times. So my wife also said that I'm not funny and don't try to infuse humor into your talk. But that being said, so we created a vision, and what we said was we would try to make starting clinical trials, science-justified clinical trials, getting to the children that needed them most as our primary mission. Papers and grants would still be important, but starting those trials and making them available to the children was job number 1. And then we would do something along the lines of what SpaceX or Thomas Edison did, bring in both people of your primary discipline, for which that was biology for us, but also engineers. And so it helps that we have 2 retired vice presidents of Intel on our board, one of whom is our executive director now, pro bono, which is very kind of him. But most importantly, we would bring families to what we did, and we would hold ourselves accountable to families that we brought these drugs to trials and they would be effective. So in doing so, we had to realize that this hadn't been done. I think the best thing about Stanford podcast series is the Y Combinator podcast series, which says that a startup is by definition something no one else believes in, no one else thinks is practical or feasible, but you have an inner belief that you can do it and you can do it better than anyone else. So we adopted the startup model to starting a nonprofit biotech. And we did a lot of things that we felt would be different and game-changing, the engineering forward aspect. We empower all of our scientists and engineers to do things that are innovative by creating a rule that 15% of everything that everyone does has to be something that myself as scientific director, I either didn't give them permission for or told them specifically not to do. And that's where the best stuff comes from. And finally, we have a flat management style, which you can do in a group of about 10 to 14 people. So we honestly did get a warehouse. So we have a 5,000 square foot paint factory where we have our BSL-2 lab and NIH-sanctioned vivarium. I will say that it was really nice that in between it being a paint factory for 70 years and us going into it, Nike took it over and remodeled it as an off-site creative space for product launch parties. So we're probably the only BSL-2 lab that I know of that has both a DJ booth and a bar. And everyone says, millennials, everyone has what they say about millennials. And I brag about my millennials. I say, I love my millennials. I want our lab to be a millennial lab. Because you can't necessarily motivate people with money if they're millennials, but you can motivate them with a mission. And I've come to realize a few things. They don't care about the 7% retirement account contribution from us as an employer without needing an employee match, which I thought would be really important. They really just care about the trail mix and pizza oven and the espresso machine. So. And they work fabulously, and they meet the patients. We have pictures all over the laboratory. You can kind of see them there in the background of families. These families come visit. Some of these families, their pictures are of children we know who are alive with us in this world. Many of them are of children who are not alive with us in this world, but their families come visit, and it has a unmistakable impact on these young scientists. So here is what our mission is. We do a very simple thing. We make it really easy for pharmaceutical companies and clinical trial networks to engage for childhood cancer. We de-risk their involvement. We figure out how a cancer works and identify a target. We do the preclinical studies that say if we had that target and a drug against that target, that it works not just in one child's cancer, but in an array of a dozen of a children's cancers. So we currently have a clinical trial that we're working towards that doesn't just have one animal model, but it has 24 patient-derived animal models. That level of scientific rigor. And the nice thing is, even though pharmaceutical companies say, we don't have the domain expertise, the money, or the time to understand the targets. We don't have the financial resources to want to invest in studying and validating every drug in a preclinical setting. But absolutely certainly, we have children and we care about children and we want to be advocates, internal champions. Since you've de-risked the target validation, you've de-risked the preclinical validation, we of course will give you $600,000 worth of drug to take into the clinic with the cooperative groups. And the cooperative groups, the Children's Oncology Group, the Pediatric Brain Tumor Consortium, they say, you've provided us such fabulous scientific data justifying taking this clinical trial forward that, and the drug company wants to give us the drug. Of course, will take this well-justified drug into clinical trials. And so, so that's what we do, and we are in our 26th month, and in our first 23.5 months, we got 2 drugs into 2 national clinical trials, one for the brain tumor DIPG, published that result in Nature Medicine, one for the muscle cancer rhabdomyosarcoma, published in Genes in Development. So the way this is possible, what's at the core of our startup, is the relationship with families. And we need those families engaged, but we also need them empowered. If they're properly empowered, they're properly educated. And so, you know, here's an example of somebody who was sitting in the lab literally 2 days ago. So this is Nathan. His daughter, Natalie, she had alveolar soft part sarcoma. He really sort of holds us accountable. He doesn't bring the axe to the lab. But this is actually the Timbers are a Portland soccer team, and so that has to do with the axis. But what we do is we say, okay, tell us what you care about. And so, for instance, Nathan was interested in understanding alveolar soft part sarcoma. So we bring 20 people to Portland for a week's boot camp, and they get to pick 2 or 3 topics that we cover. And in doing so, they learn all about the basic science investigation of targets. They learn all about the process of drug development with pharmaceutical company speakers. They learn about clinical trial designs. And then after all of and they have hands-on time in the lab. And after all of this, they write a roadmap for how to cure the disease that guides their investment, the community-based investment. So, so that being said, I think I've spoke most about these, but I'll just show you the roadmaps that they've written. And it's my rule that if you're empowered, you don't need me on the publication. You write the paper yourself. And so these are 2 publications, one on the childhood liver cancer hepatoblastoma, the other on the childhood brain tumor DIPG, published in the peer-reviewed literature by only families and high school and college students. So now there's a lot of work involved, and people will say that week is the hardest week of their life, that when you're juxtaposing a childhood cancer survivor with parents whose child didn't make it, there's a lot of emotional torment. They all say it's the best week they've ever had in their life. And this is an example of their work product. So there are things across these different rows, the top, second, third, and fourth rows. The top one is where a lot of these diseases are. You know, is there a registry for a particular disease, and does it track outcomes? Is there a biobank? If that biobank exists, is it centralized, or are there lots of little silos? Is it okay to have lots of silos if they talk to each other? Are there enough cell lines and animal models of disease that you're sure that there's enough diversity in the models to reflect the patient population? And then, of course, you know, I trained with someone who came up with the technology for genetically engineered mice, Mario Capecchi, and so transgenic animals are always close to my heart. But beyond that, there's a number of other things, genetic tests, protein level tests, chemical and drug tests that are done, integrating things. Big data is huge, and it's here. The data already exists. So much of it isn't integrated, though. And then getting the things done, providing the evidence that justifies to the pharmaceutical company to move forward with the trial. These are the steps. And in some instances, it's just at the beginning. So hepatoblastoma, there simply are not enough cell lines that represent the actual disease. But with DIPG, we're already down here. And DIPG, just to talk a little bit about this is a disease for which, as a scientist, I'm really invigorated by. I'm certain there's so much that we're going to learn, and their outcomes are going to be fabulous. But it's an awfully difficult place to have a tumor. So it's in the pons. The pons is the midbrain, and it turns out, you know, it should be about the size of your pinky. The pons should be, but it instead gets to be about the size of a sort of a walnut or a prune. And there's only so much space in the skull, and as a result, when these eventually grow, they will push the limits of the volume of the skull. The brain won't be able to compensate with the size of the growth of this tumor, In fact, the survival for this tumor is 0, and the median survival without radiation is 7 months, with radiation is 9 months. But we set about, when I was with the Children's Oncology Group as the co-chair of the New Drugs Committee, we sat asking ourselves, how can we make a difference in this disease? Now, there's sort of a challenge there, because if you're going to pick a drug for a disease that's rare, it's going to have to be your clinical trial, your go-to clinical trial for 3 to 4 years. You simply are going to have enough patients enroll, and that's going to take time. So everyone in the room had an idea about their favorite drug based on observing expression of one target or another target. I proposed a sort of a Pillsbury bake-off of targeted drugs. We actually got 14 institutions across the United States, Canada, and Europe to work together. But what really made this work was the families. Because even though it was $100,000 that started off the project, the next $560,000 came from parents and families dialing in, offering money to extend the scope of the project. And then when a particular lab, say in France, slowed down over a holiday, they would dial up and say, you know, you haven't made any progress in the last week. And they'd say, well, we're so sorry, but it's Hanukkah and Christmas. So, so they really did hold the feet to the fire. This resulted in both the Nature Medicine publication and the NIH director, Francis Collins, blogged about it. You can read Esther Landis's article in Scientific American if you want to learn more. I'd say, though, my favorite story from the Nanocourse was that of Andy. So Andy came to one of our Nanocourses, and at the end of the week, he said, I kind of like this. How do I get more involved in this? And I said, well, you probably need to see if you have the chops, if you have the hands to be able to work in a laboratory. And he said, you just need to find somebody willing to give you a sabbatical. And he says, well, when am I coming to your lab to do my sabbatical? And I'm like, well, I don't know, Andy. You know, and I'm like, well, first of all, I'm not paying for it. So he got the Alex's Lemonade Stand Foundation, because there was a representative of the Alex's Lemonade Stand Foundation grants office there that paid for his summer internship. He actually got things done. I mean, he's Stella's dad. Stella's with us in this world. But the amazing thing is this college-educated stone and tile mason who's been featured on HGTV for dream bathrooms in Big Sky cabin resorts. He retooled, he gained enough preliminary data that he actually was just awarded last week a $100,000 grant from the Raleigh Foundation to study his daughter's tumor for 2 years. So that, to me, is the ultimate level of empowerment, and he continues to empower other families. So I'll wrap up and say that you can do amazing things with empowered families, and I greatly appreciate your time and attention. So we are about 20 minutes into this, and so we have time for a few questions, or we can go to the next speaker. Yes. Right. So the cancers with the least resources have no cell lines, and sort of I think you probably more than most people would be aware. There's two kinds of science. There's the naturalist who observe things, and then there are the experimentalists who push and pull on things. If all you have are tissue samples from biopsies, you're not going to be able to really prove that anything turns a cancer on or makes it work. When you have cell lines, you most certainly can. So we try to pick things that other people aren't working on. We're rallying the families together around sending in samples is really important. And what I didn't mention, and I'll mention now, what the DIPG parents started on their own, what drives a lot of this, is that the 1 in 5 kids who won't be cured, they're hard to always know who they are when they're biopsied at diagnosis. When the patient relapses, you absolutely certainly know what it is. You don't need to poke them. You don't need surgery or anesthesia. But the only way to really understand the cancers that we clearly don't know how to treat, to make the cell lines for the diseases that we don't have cell lines for, is to obtain tissue. The only time to do that painlessly is after life. And so we have a legacy gift program that really is well subscribed, oddly, and driven by the community, not medical care providers, where they bring those families come in contact with us. We work with the clinicians taking care of the family, and when the child passes away, we reimburse the cost of moving the decedent, the child's body, from the home to the hospital, where the local hospital does the autopsy for free and sends us the material in a FedEx box that we can make cell lines and material from and then share with other labs. Okay, well, again, thank you for your attention.